Welcome to E-Content Development Center, Majalis Arts and Science College, Puramannur. I am Serena, Faculty of Department of Microbiology, Majalis Arts and Science College, Puramannur. Today we will discuss about Rickettesial infection. Rickettsia are a big family that consists of a diverse group of species that causes human infection. Rickettsia are strict intracellular parasite and transmitted through hemococcus arthropod vectors, and they are gram-negative bacilli and obligate intracellular parasite like viruses, but they are similar to that of bacteria. Some features like their cell wall is made up of peptidoglycan, they have metabolic enzymes, they have both RNA and DNA, they multiply by binary fission like bacteria, and also are susceptible to antibiotics and other antibacterial agents, and they are first discovered by the scientist Howard Taylor Ricketts, hence the name Rickettsia. Then the Rickettsia family consists of three genus, that is Rickettsia, Ornithischia and Ehrlichia. In this or among these three genera, Rickettsia is a human pathogen that causes the diseases like typhus fever and spotted fever. Then next is about Rickettsia and its morphology. Rickettsia are gram-negative bacilli, and they are similar to bacteria and exist as coccobacilli. Sometimes they exhibit pleomorphism, and their size is 0.3 to 0.6 into 0.8 to 2 micrometer, and they are cultivated only in cell-free media. Okay, next is about their cultivation or cultural characteristics of Rickettesial species. Here, Rickettsia are unable to grow in cell-free media. Their growth occur in cytoplasm of infected tissue or the spotted fever causing Rickettesial species that grow in nucleus of the infected tissue. And their optimum temperature is 32 to 35 degrees Celsius. That is the optimum temperature needed for the maximum growth of Rickettesial species. Rickettesial species that grow only inside a cell, or it is a strict intracellular parasite. There are three methods used for the cultivation of Rickettesial species, that is embryonated egg culture, tissue culture and animal inoculation. In embryonated egg culture, yolk sac is the site for the maximum growth of Rickettesial species, and in animal inoculation, guinea pig is used as a experimental animal for the growth of Rickettesial species. Here intraperitoneal inoculation of Rickettesial species is needed in the animal inoculation method. And in tissue culture, these Rickettesial species are sensitive to mouse fibroblast as well as Hep cells and HeLa cells as well as Vero cells. Only these cells are sensitive to growth of Rickettesial species. Next is about their resistance. This Rickettesial species can be inactivated by physical methods like heating, drying, etc., as well as chemicals like common antibiotics and antiseptics. They can be readily destroyed at 56 degrees Celsius and at room temperature when they get separated from the host body, because they are obligate intracellular parasite. Then next is about their antigenic structures. This Rickettesial species consists of three antigenic structures, namely surface protein antigen, outer membrane protein and alkali stable polysaccharide antigen. Here surface protein antigens are genus specific and consist, or they carry cross-reactive epitopes. That means they are cross-reactive with the other species. And second type of antigen is outer membrane protein. They are two types, A and B. They are species specific and needed for the adhesion of Rickettesial species to the host body. Then third one is alkali stable polysaccharide antigen. They are the important antigen present in the Rickettesial species. This antigen is shared by Rickettsia as well as Proteus species, Proteus like Proteus mirabilis and vulgaris. That is the basic principle of Weil-Felix reaction that is used for the diagnosis of Rickettesial infection. The third important antigen of Rickettesial species is alkali stable polysaccharide. This antigen is shared by Rickettsia and Proteus species like Proteus mirabilis and Proteus vulgaris, and the antigen present in the Proteus mirabilis and vulgaris are OX19, OX2 and OXK. These antigens are similar to that of polysaccharide antigen present in the Rickettesial species. This is the principle or basic principle of Weil-Felix reaction used to diagnose the Rickettesial disease. Okay, next is about pathogenicity and clinical features caused by Rickettesial species. Here, Rickettsia causes two types of disease, typhus fever and spotted fever. Typhus fever is again classified into three types: epidemic typhus fever, recrudescent typhus fever and endemic typhus fever. First is epidemic typhus fever. This epidemic typhus fever is caused by Rickettsia prowazekii, and it is transmitted by the insect or vector known as body loss. Hence, it is known as loss borne. And this epidemic disease or epidemic typhus fever, that is transmitted from one person to another through feces of infected fleas or through the dried feces of fleas inhaled by another person, and also transmitted through the wounds or abrasions contaminated by the infected feces. And this incubation period of this disease is 5 to 15 days, and the disease is onset with the fever and chill. And the other symptoms include skin rashes on first appeared on tongue and then spread to palms, soles, hands, fingers, etc. Next is about the next type of disease or Rickettesial species, that is recrudescent typhus fever. It is a type of epidemic fever when the or one person who recover from the typhus or epidemic typhus fever, the organism Rickettesial prowazekii or Rickettsia prowazekii that remain latent in the body of the patient, and they get reactivated and causes epidemics again, that is known as recrudescent fever. Then next type of typhus fever is endemic typhus fever. It is caused by Rickettsia typhi, and the rats act as a reservoir of this Rickettsia. Then next type of typhus fever is endemic typhus fever, which is caused by Rickettsia prowazekii, and this species are maintained in the environment by the reservoir, that is rat. Rat act as a reservoir for the maintenance of this Rickettesial species in the environment, and this infection is transmitted from one person to another through feces of infected fleas by bite of the fleas, as well as through ingestion of contaminated food and water, and this infection is milder than that of epidemic disease. Then next type of disease caused by Rickettesial species is spotted fever. Here spotted fever is again classified into two types, Rickettesial pox and thick typhus. Rickettesial pox is caused by the Rickettesial species known as Rickettsia agaricus, and the domestic mouse is the reservoir and the vector is mites. And this Rickettesial pox resembles chicken pox, hence the name Rickettesial pox, and this is also known as vesicular or varicella-form rickettsioses, and this is a self-limiting disease of humans and is first observed in New York in 1946. The second type of spotted fever is tick typhus, and this tick typhus is transmitted by the vector known as ticks, and these ticks again act as a reservoir of Rickettesial species that causes spotted fever, that is tick typhus. And this tick typhus is again classified into two types, Rocky Mountain typhus fever and Indian tick typhus fever. Rocky Mountain typhus fever is caused by Rickettsia rickettsii, and it is the mildest form of spotted fever, and Indian tick typhus fever is caused by Rickettsia conuri. That's all about the first part of rickettsial infection. Continuation of this disease we'll discuss later.